Okay, ma'am. Thanks a lot. So, very good evening to one and all present over here. I would like to appreciate the organizer for giving me this opportunity to present my research work on flaxseed mucilage, flaxseed mucilage and HPMC, as well as sodium alginate polyvinyl alcohol composite bilayer film as a promising carrier for the periodontal treatment. Currently, I am working as associate professor at JSPMs College of Pharmacy and Research, Tathode, as ma'am have introduced. So I'll start with my presentation. This is the content of my presentation. So periodontitis, if you see, periodontitis, basically it's a chronic all over the world, and it is one of the gum diseases, and it is mainly caused by the bacteria such as periodontal or P. gingivalis or AAP or P. fortastus, etc. That mainly damages the soft tissue around the teeth, and without the treatment, periodontitis can also destroy the brain or the bone that support the teeth. So it can cause the teeth to loosen or to that may lead to the tooth loss. It is characterized by the loss of soft tissue attachment and the alveoli bone resorption that is caused by the pathogenic microorganisms that will result in the pocket formation or the gingival recession. So it's one of the six cause or chronic cause of the diseases. This is the pathology, how it proceeds inside the gums. So if there is absorption of salivary peptidase or the glycan proteins, these proteins will be deposited over the teeth that may form the calculus or that may form the tartar. Then the tartars mainly attracted towards, or the bacteria mainly attracted towards this tartar, and there is a calcification of this plaque. Then bacteria, when they feed on this tartar, they will generate certain enzymes and endotoxin or the endotoxins that will mainly responsible for the inflammation, and such inflammation go on progression, and finally it will responsible for the loss of teeth, and that may be very dangerous, especially in the small kids. So these are the general treatment strategies for the treatment of periodontitis. Most of the times in severe cases, people mainly refer the surgical treatments, and some non-surgical treatments are also available at the initial stages, like scaling, root planning, and topical antibiotics. Scaling refers to the cleaning of the tartar. Root planning refers to the smoothing of the surface of the teeth, and this is our area of interest, that is the treatment with the help of antibiotics. Surgical treatment involves the flap surgery, which mainly reshape the muscles or the gums. Then there may be a generation of soft tissue graft, where the muscles from the upper palate they are extracted and they are grafted over the gums. Then bone grafting is also there when there is a loss of bone, or when the almost there is a degeneration of bone. In such cases, there is a bone grafting. Then guided tissue regeneration is there, where mainly dentist keep certain kind of material under the gums, and because of that there is a specific growth of gum cells. And next one is the tissue simulating proteins may be used for the as a surgical aid. So this is of our interest, where we can target or specially target the periodontal pocket, or the area between the teeth and the gum, so that we can have the n number of options, like the gels or the films or the strips, which can be easily attached in this particular section. But the major limitation is that we are having very limited area where we can keep our products. So product has to be designed very preciously, so that you can have the optimum utilization of dose, as well as we can give the or your dosage form should fit within this small cavity. So my aim of our research work is to develop the flaxseed mucilage HPMC and sodium alginate polyvinyl alcohol composite bilayer film as a promising carrier for the periodontal treatment. So objective is to formulate the mucoadhesive films that are mainly based on the natural origin, and flaxseed mucilage and HPMC films, it mainly contain the drug doxycycline hyclate, which is the major antibiotics used in the treatment of the periodontitis or the gum diseases. And next one is next layer is made up of sodium alginate and polyvinyl alcohol, and it mainly comprise of the clove essential oil. Clove essential oil mainly contains the eugenol, and that is having the anti-inflammatory or the antibacterial properties, and it has been proved since many years that it is having anti-inflammatory properties. So whatever rationale I have already explained in my aim and objective. So both flaxseed and HPMC, they are having the mucoadhesive properties. Uh, so if we go through the structure of the drug, I am going to focus on the drug, or I had focused on the drug doxycycline hyclate. It is highly soluble in water, 100% bioavailable. It is having the log P of 0.63. That means it is highly suitable for the development of dosage form. It is having half-life of 41.3 to 41.6 hours, and it is having it belonging to the BCS class 1. So it is highly soluble and highly permeable also. It is having the Cmax of 1.7 mg per liter and Tmax of 2.5 to 3 hours. It is having pK of 3.09, and it can be stored at in the temperature range between 0 to 6. But at such, it is also stable at room temperature. Next, my focus or the next layer contains the eugenol that is taken from the clove oil. It's a pale yellow color of liquid, and it is having quite traditional acceptance for the treatment of gum diseases, and what we can say, it is miscible with alcohol, but it is insoluble in the water. Why I have chosen the doxycycline hydrate? Basically, it is having or it is having the effect on MMP activity, that is the matrix metalloproteinase activity. That is associated with the calcium and zinc binding properties of zinc binding properties of certain enzymes, as well as it is also responsible to degenerate or to interfere with the 30S ribosomal units of the bacterial cell. So it will interfere with the formation of bacterial cell also. And next one is the eugenol. It is effective on both gram positive and gram negative bacteria, and basically it acts on the cyclooxygenase 2 enzyme to initiate the anti-inflammatory action. I have chosen the flaxseed. Generally, it is available into two varieties: golden yellow and the brown flaxseed. Brown flaxseed is readily available in nearby markets, so I have selected brown flaxseed as to extract the mucilage. This is the authentication certificate from the Botanical Survey of India. If you go through the general properties of the flaxseed, it contains mucilage, and mucilage is of two types: acidic polysaccharides and the natural polysaccharides. It is highly soluble in the water, pH is almost neutral, and mucilage is available as amorphous brown colorless powder. These are the content of other polymer that I have used: hydroxypropyl methyl cellulose, JSPMC, sodium alginate, and PVA. These are the molecular structure, molecular weights, and all these polymers are soluble in the water except PVA, that is not soluble in the cold water. It has to be heated and then get it solubilized. Yes. So why I have selected this polymer? Simply because they are readily available. They are they have proved properties of the biodegradativeness, and PVA is also famous for its disintegration or immediate release properties. This is the plan of my work. Literature procurement of drug and extraction of or procurement of flax seeds and extraction of mucilage. I have performed certain formulation studies, and I have formulated the double layer film, and that double layer film was further evaluated. So extraction of mucilage, I have taken the flaxseed near about 50 grams. It has been soaked in the water for minimum 12 hours. Then it was stirred at I guess, yes. It was stirred for about 100 RPM for 3 hours and heated up to 80 degrees centigrade. Further, it was centrifuged, and whatever centrifusion mass is there, that was separated, and finally ethanol was added to extract the precipitated mucilage. Mucilage was further dried in an hot air oven, and it was found that almost the yield was about 14%. The pH of mucilage was 6.94, which is near about the reported value. It is having high swelling index near about 83 to 83%, Ash content is 4%, acid insoluble ash and water soluble ash is 0.2%, and it is highly so swellable in water and insoluble in ethanol. As I have used the ethanol as a solvent to extract the mucilage, it indicates that it is highly insoluble in the ethanol or other organic solvent. LOD is near about 1%. It shows the positive test for the carbohydrate reducing sugar or monosaccharides, mucilages. The proteins and amino acid were found to be absent. Next was the preliminary studies for the eugenol. I have to confirm whether my sample contains eugenol or not. So for that, I have run the TLC, and the reference of the observed value of TLC is near about the reported value of 0.37. Okay. Oh, sorry, the 0.32. This is the calibration curve of doxycycline nitrate, whether it will show the lambda max near about 271.93, almost 272 nanometers. And this is the calibration curve of doxycycline. I have done the calibration curve in the phosphate buffer 6.8. Next one is the calibration curve for the eugenol. It showed the maximum wavelength near about 281.54, and it is not soluble in the water, so all the dilutions are made in the ethanol. This is the graph or the IR spectra for the drug excipient compatibility studies, where it show your first graph shows the pure doxycycline hyclate. Next one is the physical mixture of doxycycline hyclate, HPMC and flaxseed. Third one is the formulation of the primary layer D1, which contains the composition of flaxseed as well as the HPMC along with the doxycycline. This graph mainly shows the graph of clo oil or the pure clo oil, and last graph is for the formulation of sodium alginate and PVA layer containing the clo oil. All these graph shows no extra functional group, or not a single peak was diminished. Certain peaks are broadened simply because of the physical interaction that indicate the compatibility between the drug and excipient. Next one, I have prepared the primary drug loaded layer, which contains the HPMC and the flaxseed mucilage. So I have taken the 1:1 proportion of HPMC and mucilage, or I have taken the in this particular table, I have taken the flaxseed mucilage into the different concentration, and the concentration of HPMC was constant. And I have used the glycerin as an plasticizer. So when I have prepared film of, or when I have gone through this kind of composition, F1 film that contains flaxseed mucilage, that doesn't show formation of film. That means pure flaxseed is too sticky to form the film. Then F2 films that contains equal amount of flaxseed mucilage and HPMC, it showed the perfect formation of the film. The film was not sticky and the thickness was optimum. Then F3 film contained 300 milligram of flaxseed and 200 milligram of HPMC, but the film was too sticky and it is less flexible, or it is not easily peelable. So as we go on increasing the concentration of the flaxseed, the films are quite sticky or they are not peelable. So at higher concentration of the flaxseed, the films are not formed. So 1:1 ratio of flaxseed to the HPMC was selected to optimize the primary layer. Yes. Now there is a formation of the secondary loaded film, or the film containing the sodium alginate and PVA. So I have used the different concentration of sodium and alginate and PVA, and glycerin was used as the plasticizer. Clo oil was dissolved in the ethanol and twin 80, and further it is added into the polymeric solution to cast the film. So these are the different composition of sodium alginate and PVA to form the secondary layer, and they are made, the final volume was 10 ml. So in this also we have found that 1:1 ratio of PVA and the sodium alginate give the optimum film that is having the optimum flexibility, transparency, and easily peelable. So further, these films are evaluated individually and bilayer films are formed. So they are evaluated for thickness, folding endurance, and disintegration. Formulation of the secondary layer, or formulation of the secondary film, it contains the different composition of sodium alginate and polyvinyl alcohol. So the folding endurance was range in between almost 190 to 209, Then film thickness. Folding endurance was calculated by repeatedly folding the film at the same point again and again till it breaks. Then film thickness was calculated by the vernier caliper. It is ranging from 0.21 to 0.28. Then weight uniformity, the films were weighed and the average weight were taken. Disintegration time, the films were kept into the phosphate buffer and the time to disintegrate the film was noted, and it was ranged between the 6 minutes to 12 minutes. Okay. So the optimum concentration of 1:1 ratio gives the better results, or they are quite giving the promising result. A percentage was regional content was calculated from all these different batches from the secondary layer, and it was found that again 1:1 ratio of sodium alginate gives the almost 92% of the regional content, and it was mainly done by taking the area of 9:9 square of film and dissolving it to the phosphate buffer, and finally absorbance was taken at 281.54 nanometer. The drug release from the secondary layer was also estimated, and it was found that the maximum drug release from the secondary layer was 68, almost 68%. Rest of the layer, though the concentration of polymer was higher, the amount of the drug release was lower. Okay. So this is the lowest concentration of polymer, which gives the highest amount of drug release within 10 minutes. And drug release was basically calculated or basically carried out in the 100 ml of phosphate buffer, and the sample was taken for maximum 10 minutes. This is all with respect to the evaluation of secondary layer. Next one is the evaluation of primary layer, that contains the 1:1 ratio of HPMC, and it is incorporated with the 40 mg of doxycycline hyclate. The primary layer was first formulated, and then this is the primary layer which contains the flaxseed mucilage and HPMC, 40 mg of doxycycline, and glycerin was used as a plasticizer. And over the primary layer, the secondary layer was loaded, which contains 1:1 ratio of sodium alginate and PVA, which is loaded with the 10 mg of clo oil. So dose calculation was done for the doxycycline hyclate. So depending upon the diameter of the petri dish, we have calculated the area of the petri dish, and from area of the petri dish we have selected the area in which we are getting almost 40 mg of the doxycycline hyclate, or almost 10 mg of eugenol content. So based upon the area of the petri dish, we have calculated how much amount of expected drug should be present within that particular area. So almost 9:9 mm square of the film contains 40 mg of drugs, and almost 9:9 mm square of the petri dish mainly contains the 10 mg of eugenol. So this is the method to prepare the bilayer film. I have prepared the first primary layer. That primary layer was coated with the 5% of calcium chloride solution, and over that there is a loading of secondary layer. So primary layer mainly contains the flaxseed mucilage and HPMC. Those polymers are heated. Drug is added to form the film, and that the film was further sprayed with the 0.5% of solution of calcium chloride. And over that solution, I have just casted the secondary layer containing the sodium alginate and PVA. The drug contained from the primary layer as well as the bilayer film was evaluated, and it is almost 98 and 97%. That is within the limit of pharmacopoeia. Folding endurance is much more higher. Film thickness was ranged from 0.21 for the primary layer, and it was quite higher for the secondary layer. Weight of the film was found in between 0.143 for the primary layer and 0.285 for the secondary layer. Disintegration time for the primary layer was almost or both the layer was almost 8 hours. Okay. Swelling study was carried out by placing the films into the phosphate buffer solution pH 6.8. The films are soaked in the buffer, and later on periodically the films were removed. They were or the whatever buffer was there, that is removed off with the help of tissue paper, and then they are revealed. So this is the data for the swelling studies. Both the layer, that is primary layer containing the flaxseed mucilage and HPMC, as well as the bilayer, they are having the highest or higher swelling capacity. But the bilayer film is showing the more swelling ability as it contains two more polymers in addition to HPMC and the flaxseeds. This is the surface morphological study for the primary as well as the secondary layer, where it shows the distinct formation of the two different layers. Okay. So this is the oil loaded film without addition of 0.5% of calcium chloride. The film was detaching, but when the calcium chloride solution was sprayed, the uniform bilayer film was formed. These are the scanning electron microscopic images indicating the intactness of the film. Though the film surface was rough, but it is free from the crack. Then tensile strength was determined is analyzed to determine the mechanical strength. It was carried out by the Brookfield tensile strength tester, and it was range in between 411, or for D1 or primary layer it was 4.11 newtons centimeter square, and for secondary layer the mechanical strength is higher than your slightly higher than the primary layer. Then bioadhesive force was also determined with the help of texture analyzer Brookfield Engineering Limited. For that, goat mucosa was used as an mucosal membrane or the model ex vivo membrane. So even both the films are there, they are possessing the better bioadhesive strength. Primary layer is having the lesser bioadhesive strength as compared to the bilayers. In vitro drug release studies was carried out by USP type 2 dissolution apparatus by using the phosphate buffer 6.8. The volume was 100 ml, and the samples were removed for a minimum period of 8 hours. So this is the data for the dissolution study for the bilayer as well as the primary layer. Bilayer shows the more sustained release as compared to the primary layer. Almost 82% of the drug is released from the primary layer up to within 6 hours. Okay. Antimicrobial test was carried out by the cup and bob method, or you can say by using the Kirby biodiffusion method, where Muller-Hilton agar was used as a standard agar, and the wells were prepared by 7 mm diameter. Okay. And your samples were applied in the MHA wells and incubated for 37 degrees centigrade for 24 hours. It was carried out for two bacteria, that is Staphylococcus aureus and E. coli. Staphylococcus belonging to the gram-positive bacteria, and E. coli is belonging to the gram-negative bacteria. So to check the effect on both the bacteria, this kind of test is carried out. So these are the results where a plain doxycycline layer that will showing the, or that will exhibiting the effect on the E. coli. This one is the plain doxycycline layer. This one is the plain eugenol loaded film, and diameter is almost near about or above 16.4. Okay. Then this is for E. coli. Next one is the effect of the plain doxycycline on the S. aureus, that is having the diameter of 12.1 mm, and effect of eugenol on the S. aureus, that is having the diameter near about 11.5. This indicates that the eugenol or the doxycycline hydrate, they are having the more profound effect on the gram-negative bacteria, and all the bacteria that is responsible for the periodontitis, they are having the major effect on the, or they are majorly belonging to the category of gram-negative bacteria. This is the actual results of the bilayer film. So bilayer film was tested on the E. coli, and it is showing the zone of inhibition about near about 22.5, whereas bilayer film on the gram-positive bacteria S. aureus having the zone of inhibition near about 20.8 mm. That means both bilayer film is having excellent activity against both gram-positive as well as the gram-negative bacteria. So the conclusion of the study is that prepared films will be used as a potential therapeutic option for the management of periodontitis. Further studies must be carried out on the to access or to examine the in vivo potential of this bilayer film. Yes. These are my references, and I'm again thankful to organizer for providing me this precious opportunity and webbing